Hello everyone. Today we are going to study about the important topic related to respiration of plant. Uh, Generally, a student have a problem on glycolysis. Uh, this is the first step for the respiration in plant. Glycolysis is the common step for aerobic respiration as well as anaerobic respiration. Aerobic respirations means the respiration in which O2 is present is required in aerobic respiration. Uh, uh, Whereas in anaerobic respiration, O2 is absence or in a less sufficient amount. In aerobic respiration there is sufficient amount of oxygen required. In glycolysis, what happens here that the glucose having a structure like this, here oxygen, there are 6 carbon. Glucose is 6 carbon compound, and this glucose which we have got from photosynthesis of plant is utilized in respiration, and in respiration we generally think of oxidation, but in glycolysis there is not oxidation of carbon dioxide. There is partial oxidation of carbon dioxide, because in glycolysis glucose is converted into a pyruvic acid. So there are various steps. In first step there is production of pyruvic acid, 2 mole of pyruvic acid. So in glycolysis the plant can utilize glucose either fructose, both are 6 carbon compound, and from 6 carbon compound glucose first get converted into glucose 6 phosphate, then glucose 6 phosphate is converted into fructose 6 phosphate, then the fructose 6 phosphate is converted into fructose 1 6 biphosphate. Then this 6 carbon compound, all those are 6 carbon compound, and this 6 carbon compound is get splitted into 3 carbon compounds. The first one is 3 phosphoglycerol aldehyde, and another is dihydroxy acetone 3 phosphate, dihap we generally say, and they both are isomer of each other. Then dihap can be converted into 3-PGL, and 3-PGL can be converted into dihap. So we will get the product, one pyruvic acid from one side and another pyruvic acid from another sides. So we generally will do the only one pyruvic acid, and then we will multiply by 2. So 3-PGL will be converted into 1 3 1,3-phosphoglycerol aldehyde. Then 1,3-phosphoglycerol aldehyde is converted into 1,3-phosphoglyceric acid, and 1,3-phosphoglyceric acid is converted into 3-PGA phosphoglyceric acid, and 3-phosphoglyceric acid is converted into its isomer of 2-PGA phosphoglyceric acid. 2-phosphoglyceric acid is converted into 2-phosphoenol pyruvate, and the last product which is required, which we get at the end of the glycolysis, is pyruvic acid. We can even say pyruvate too. So from here the compound are multiplication of 2, means 2 mole of every compound are formed. So we will get 2 mole of pyruvic acid at the end of the 2-pyruvic acid in the end of the glycolysis. So generally in net reaction, we will input here in first step. In the first step we enter the ATP and we get ADP. In first step 1 ATP we input the 1 ATP and we got 1 ADP. ATP is a triphosphate and ADP is a diphosphate. One phosphate is added in a glucose to form a in the sixth carbon compound, in the sixth carbon of the glucose. So glucose 6-phosphate, the name is given. And in this step, this is the isomerism step, means there is isomer, both are isomers of each other, and this is the first step, this one is the second step, this one is the third step. In third step we add another ATP, and we get ADP. Why we did add ATP? Because it is 3 carbon compound, 3 phosphate, ATP is triphosphate, 3 phosphate, and it's converted into diphosphate, means 1 phosphate is utilized, and the 1 phosphate is get utilized in a fructose 6-phosphate in the first carbon of the fructose. So phosphate is attached with first carbon of the fructose, so fructose 1,6 biphosphate, because there are 2 phosphate. One was already present there in the 6th carbon of the fructose, and the another one is added to first carbon, so it is altogether 2 phosphate, so biphosphate, fructose 1,6 biphosphate. And here the enzyme is used, aldolase, to split into 3 carbon compound. This one is 3 carbon compound, this one is also 3 carbon compound, and both can be interconvertible by a enzyme called triose phosphate isomerase, because since there is isomerism is occurring, so isomerase enzyme is used. Generally the enzymes are name is given on the basis of the product is formed, from which the product is formed, what is the step related to it. Since it is isomers of each other, so they are called isomers enzyme, and there is a 3-PGL is a triose phosphate. This is a 3 carbon compound, triose. So triose phosphate, because there is phosphoglycerol, there is phospho. So from the name itself we can find the enzyme name. So it is the isomer enzyme, and this is the fourth, this one is the 5th stage, and the 5th is in the fifth 3-phosphoglyceraldehyde is converted into a 1,3-phosphoglyceraldehyde, that is addition of phosphate as 3PO4. It is dead, but there is no enzyme is required, so this step is the non-enzymatic step. Non-enzymatic step. Remember, fifth step of glycolysis is non-enzymatic, but all other are enzymatic action step. So basically in fifth step, the 3-phosphoglyceraldehyde is added the phosphate as 3PO4 inorganic phosphate, then it forms the 1,3-phosphoglyceraldehyde. This is also 3-carbon compound, and the 1,3-phosphoglyceraldehyde is get oxidized in this step. This is oxidation, and oxidation NAD plus is given, and the output we get is NADH. Since it is aldehyde and aldehyde is converted into acid, there might be oxidation, so this step is oxidation step. And 1,3-phosphoglyceraldehyde, from here 1,3-phosphoglyceric acid, the one phosphate is removed, means one phosphate is removed, but the one phosphate is added to one diphosphate to form triphosphate, adeno triphosphate. So ADP is converted into ATP, because the one phosphate is added to ADP to form ATP. So here is the 2 molecules, so there will be 2 molecule of ATP into 2 molecule of ATP, and here it is 2 molecule of NAD plus, so here should be 2 molecule of NADH. Remember, at the last I will gonna calculate the number of ATP produced, number of ATP utilized, and the net production of NATP or ATP. So we have already studied 6th step, 7th step, and in 8th step this isomer 3 PGA can be said isomer of 2 PGA. So generally isomers we can also use isomerase enzyme or mutase enzyme. So in this step there is mutase enzyme, and there is not utilization of any ATP or ADP, any kinds of energy utilized or evolved. So and the 8th step is isomerization step, and the 9th step, the 2-phosphoglyceric acid is converted into 2-phosphoenol pyruvate by evolving 2 molecule of water in the presence of enzyme enolase. Is the 9th step, and the 2-phosphoenol pyruvic acid is converted into pyruvic acid. In this step the phosphate group is released. When phosphate group is released, it is utilized by ADP to produce ATP. Since there is 2 molecule, we multiply by 2, so there is 2 ATP, here it is 2 ATP, here is 2 NADH, so at last 2 mole of pyruvic acid form. So the generally most important MCQ question may be asked in glycolysis, that how many number of ATP is used. So in glycolysis there is 2 molecule of ATP is being used in the first step and the third step, because there is 1 ATP, there is 1 ATP, altogether 2 ATP is used input, and the output we got is altogether 2 ATP, we get 4 ATP output. So the net ATP production is 2 ATP. So and 2 NADH is also produced in this step and not even utilized for the any production any utilization. So it is only produced NADH. So 2 NADH is produced, 2 ATP is produced, and at last 2 pyruvic acid is produced. 2 pyruvic acid is produced from, all these are produced from one molecule of glucose. This is the final conclusion for glycolysis. And I must share with you all that the first, third and 10th step are irreversible steps and all others are reversible. And whenever there is production of ATP and NADH, it is oxidation. Whenever there is production of ATP and NADH, it is oxidation. So there is production of NADH in this step, there is production of ATP, it is also oxidation, this is also oxidation, there is oxidation too. So there are oxidation, there is ATP synthesis. If there is ATP synthesis, if there is NADH is produced, this is oxidation. So the glycolysis is also known as EMP pathways. EMP pathways, because it is the name given by discovered by the scientist Embeden Meerhof Parnas. Embeden Meerhof Parnas pathways, and there are total 10 steps in glycolysis, and there is no any carbon dioxide is evolved, so there is no carbonylation reaction is occurring in this glycolysis. And most important things, the glycolysis takes place or occurs in cytoplasm of a plant and occurred in both prokaryotic and eukaryotic cells. And thank you for your supports. Thank you very much.